All right, so we're going to continue on with our conversation about systematics. This will be part 2 of the lecture. So each part's going to be about 15 minutes. So just mentally keep that in mind, make it easier to budget your time. So part 2, we talked about basic systematics, phylogeny. We talked about biodiversity in part 1. And we left off with this idea of cladistics, the methods, kind of the methodology that we use to create those phylogenies. So when we're talking about classification, let me get a box here. What we're looking at is classification based on common ancestry. So cladistics is trying to understand this, and we organize things, again, based on this common ancestry that you're related because you have this ancestor that had these features or these genetics, these genes, etc. So think about 23andMe or those DNA tests you can do online. That's exactly what we're talking about here, except it's a smaller picture. Like, oh, I want to understand my family tree 3 generations ago. Cladistics? Well, we're talking about maybe 3 million years ago, or 20 million years ago, or 100. So same concept, just kind of a bigger picture in some cases. So we're basing it on common ancestry. Now, to determine common ancestry, it's determined by shared and derived characteristics. So, and I'll get into these in just a minute, but kind of like, what's that stuff all about? So we look at characteristics, and traditionally we were looking at physical features, length of a wing, the size of a beak, the thickness of the bone in your head, etc. Physical characteristics and features. What do we have available? A lot of that was fossil evidence. So we would organize things based on fossil evidence. Now with molecular data available, we're using that as well. We're adding tools to the toolbox to understand all of these relatedness between species. So again, cladistics and the methods we use are changing. As new technologies develop, it's phenomenal. We're changing our view of things. So when we're looking at, wow, is this related to this? If it's living stuff, we look at the molecular sequence. The DNA analysis is a huge thing. So when we're talking about viruses, technically not alive, and we'll get into that later, but we're looking at, wow, is this virus the same as this one? Are these viruses related? How will these viruses impact humans, etc.? So we're looking at, again, these characteristics and traits, typically physical features, but now molecular features are becoming very, very important. So then the data is collected about the traits, and usually these are phenotypic traits. So you guys remember a little bit about genetics, phenotype, physical features. What can I physically see? That's often what is being used to determine this relatedness. So, okay, but what we now want to do is say, okay, what is a shared trait versus a derived trait? Or what's an ancestral trait versus derived trait? So shared, let me go back up here for briefly. Shared and ancestral, we're going to consider the same. And I'll probably use ancestral more than shared just because it's more common. But what is the difference here? So, okay, so let's compare this. Is this an ancestral trait or is this a derived trait? Now, ancestral trait, I'm going to make some room here. Sorry. All right. Ancestral trait is found across all members being studied because you have a common ancestor. A derived trait is something unique. Unique to a specific group. All right, so let me make some room. I want to look at the relatedness between these three species, these organisms. So we got an iguana, we have a squirrel, and we have a goldfinch. All right, did you think about anything that could be found across all three groups or all three species? That would be an ancestral trait, something that all of them have. Now, why would they all have it? Because they share a common ancestor. So let's, again, think about this. I want you guys to hypothesize, come up with a guess. There's no wrong guess. There's unrealistic. But let's take an educated guess. What is a physical characteristic we can see in all three of these? And there's a bunch of them. So as a group, you guys could all come up with different characteristics. That's what we want. Everybody to come up with a different feature or a different group of features. And then we use those to make this comparison. So the feature I'm going to use, let me get my pen out here. My ancestral feature, lungs. All three have lungs. Reptiles have lungs. Mammals have lungs. Birds have lungs. Lungs would be an ancestral feature. Why do all three of these have lungs? Because they share a common ancestor millions of years ago that gave rise to what we call tetrapods, four-legged animals. So the evolutionary tree would do something like this. And you'd go, okay, lungs, there's a trait right there. Sorry about the drawing, it's hard to do with the mouse, or the writing I'm trying to do. Okay, so lungs is a trait that would have arose in the ancestor and then been given to all these descendants here. That was a trait that was retained. That was adaptable. That was a fit trait. But now we want to say, okay, well, what's derived? What's unique? And there's, again, lots of answers. There's no one right answer here. So a derived trait, hair. Our squirrel has hair. So we'd look at it and say, okay, they all share a common ancestor that had lungs. All three of these branches have lungs. But the reason why the squirrel, I'm going to use a red star for a squirrel, goes over here is because this new trait of lungs evolved in this group. I'm sorry, mistake there. A new trait of hair. Looking back at my writing here. The new trait of hair evolved in this evolutionary line. This line didn't evolve hair. Maybe we'd look at a different trait and say, well, what trait would be present here? What trait would be shared by this group and this group, but not shared by this? Oh, that could be something like scales. Birds and reptiles have scales. For those not familiar with it, look at the leg of a bird. It has scales on its legs. Feathers. Up here, scales here. Scales all over the body. So that would be a trait that evolved in this group, but not this group. This group evolves hair, this group evolves scales, this ancestral group. Now what's derived in birds that's not found in reptiles? Let's put the circle here. That would be something like feathers. So why are birds different than reptiles at this point? Because of the feather. That's not the only trait. Oh, that's a baddie. but that's definitely a unique feature. A derived trait or a unique feature for this group are the presence of feathers, right? So this is how this works when we're trying to look at relatedness and figure out who's related to whom and how are all these species connected. So what we're going to do is take a look at the tool known as a cladogram that we use to actually do this. So the cladogram is an awesome tool. So let's write that up here. This is a great tool that can be used to understand relatedness, to actually build that evolutionary tree, that phylogeny we discussed earlier. No, okay, so the way this works, you pick the species you want to work with. You pick the traits you want to discuss. And I encourage everybody to pick different traits. You know, you can all work with the same group of species, but pick different traits. That way you have a robust amount of data to look at. You don't want just 2 traits and everybody look at the same 2. No, we have 6 different traits across the top here. Everybody else should pick 4, 5, 6 other traits. There's literally thousands of traits. Those of you excited about molecular, go after the molecular trait. Let's look at this gene, this piece of DNA, that molecular sequence, and use all of that data. And then we take this cladogram and every other cladogram that's being used to analyze the relatedness between all of these species, and we put all that data together to get a bigger picture to understand what is the relatedness here. So, okay, so build out your cladogram and then just make this real simple, 1s and 0s. If you have this derived character, this unique trait, you get a 1. If your group, if you don't have it, if it's ancestral, you have a 0. Okay? So, it's kind of like the cooking show. We could fill this out, or, boom, here's what it looks like when it's filled out. So, lampreys do not have any of these traits. Lampreys don't have jaws, lungs, amniotic membrane, hair, no tail, bipedal. So lampreys score a big fat 0. That's okay. They're still a cool fish. Still an important part of the ecosystem. Sharks only have 1 of these. Salamanders have 2 of these traits. Lizards have 3. Get the idea? Tigers have 4. Gorillas have 5. And humans have all 6. Now, the reality is it will never be this clean and easy where you go 1 through 6 from top to bottom. It's going to be all across the board. But this simplifies what we're looking at. And then what we do is we set up the evolutionary tree and we just start drawing the lines out. Okay, so if you have a 0, you go this way. If you have a 1, you go this way. 2, 3, 4, 5, and finally 6. Okay? The higher the scores, the closer the scores are to each other, the closer the relatedness. 6 and 0 is a pretty big difference between a human and a lamprey. Gorillas to sharks, pretty big difference. But this gives us the evolutionary picture of how these things evolved. Lampreys evolved first, then this trait shows up of jaws, that leads to our sharks, then lungs evolve, that leads to salamanders, then amniotic membrane leads to lizards, and so on as we move up the evolutionary picture. So the derived characters, those unique traits, go at the branch points. Branch point is just where you have a fork in the road. And that helps us figure out when things evolved and differed, but then also when those species shared common ancestors. All right, so in part 3, we're going to talk about the cladogram and this thing known as a clade. So click on the link that says part 3. We'll go check it out.